The Panvirushield project has developed automated software technology to support government centres for disease control that deal with highly pathogenic viruses. In Stockholm, the Biosecurity Category 4 laboratory at the Swedish Institute for Communicable Disease Control is one of the few laboratories that can work with the most highly pathogenic of viruses in Europe. Virus samples are prepared for imaging using transmission electron microscopy for identification of the virus. Samples are inactivated and stained using simple preparation protocols. The method uses cheap, off-the-shelf reagents that are stable at room temperature. The procedure is straightforward and fast. The whole process takes around 15 minutes. Electron microscopy has been used for many years to study the morphology of viruses and is a well-established technique. It is the only method even today that can be used to detect emerging viruses. Sometimes viruses can jump from infecting one species to another. HIV was a disease of primates before becoming a known human pathogen. The importance of ocular TEM analysis as a diagnostic tool in virus outbreaks was proven in both the SARS pandemic and the human monkeypox outbreak in the US in 2003. The tiny sample grid is placed in the holder and inserted into the electron microscope. This open view method is able to detect interesting structures in the sample regardless of whether they are known viruses or new ones. Each square is 37 micrometers across. An average-sized particle, like this machupovirus, is 120 nanometers in diameter. The local neighborhood of each pixel in the image is analyzed, and statistical methods are used to find objects of interest. These objects are then measured and thin objects are removed. The program will then measure the texture of the remaining objects and create a property profile for each of them. These profiles are then compared with a large number of profiles in the database to find a good match. This whole analysis process takes between seconds and a few minutes to perform, instead of the hours that can be spent looking for particles by a microscopist. And in fact, identification can be made without a virus expert being present. Finally, the software produces a report that provides information needed to be able to make a diagnosis.